Tiago Cardoso, who's a senior scientist with us at Different Healthcare. And our topic is a bit interesting, and we're trying to bring quite a few things together. What would a healthcare company working with a data analytics, geospatial company, and one that works with genomics work together? The title of today's session for this afternoon is Insights on Non-Human Genomic Sequencing. So I'll start with the first bit of the title. Rashad, what do we mean by insights? Just the word insights when we talk about, because everyone here is working on AI and data. And what do we mean about insights here?
So I'll use the example of what we did last year, with the fish, right? So normally what happens is, it's like you have huge water bodies. They go, keep on trying to see how much fish is there. How do they do that? They go in huge trawlers, pick up a lot of fish in multiple locations, make an estimate of what is present in the waters, which species are present and what is present in the waters, right? So now you see that it's a huge task. At the end of the day, they get some statistics out of that. Now, if you have to use alternative data sets, right? If I'm using alternative data sets, it can be non-inclusive. I don't have to catch a fish and kill a lot of fish to get that number. It's non-inclusive. I get some data and make some proxy observations and come up with a very close statistic of how much fish population exists in our waters, and what type of fish, and what is the health of that fish that is there, which is a very crucial information, right? So these numbers we are generating from the data, the proxy data or whatever data we are collecting, is the insight in my view, basically, right? I take it at that level, then now we use that information to build on that, right? Now I collect data from satellites, I collect data from underwater sensors. I know how an environment downstairs down in the water looks like. Where is my reef? Where are other habitats? Where is seagrass growing? Where are the mangroves? This is all information I'm getting from something else, right? Now if I put all that information together with the amount of fish observations I have, so this is one observation that is already an insight. Now if I'm putting all those things together, now I know which regions in the water are helping fish to breed more, to generate more fish, for instance, right? And this tells us information on which areas we need to really protect, where this where the fish which are going extinct or going around, which areas should be protected and all those things, right? That's another level of insight. You have one insight which is coming from the initial information. Now when you're fusing multiple sources of information which are completely complementary to each other, you're getting a second level of insight in terms of mitigation, operational services and all those things.
So that's a lot of information. So, and what does that have to do, when we look at the second part of this talk, we say non-human genomic sequencing. So Tiago, is there such a thing as non-human and human genomic sequencing? And what's the difference between the two?
Good afternoon, everyone. Human genome sequence, all of us here, right? So no human genome sequencing, I'll take my notes to explain to you guys. Everything related to food, microorganisms, plants, animals, we consider no human, right? So sequencing all of that, understanding all of that, applying AI on top of it can help government to take better decision how to maneuver, how to administrate that. For example, taking the example of Dr. Prashania fish, last 1 year and a half, we have been monitoring the health of the environment in the oceans using boats who goes out in the ocean, collect water, we filter it, and we sequence that water per se. That water has the DNA of all bacteria, all protozoa, all viruses, and also the fishes who spread the DNA out in the water. So we are sequencing all of that, gathering data out of it to make informed decision to the government how to administrate the resources of the environment, looking into preserving and conservate the nature.
it's a strange place to be sitting on GIPEX, there's a lot of AI geeks here, people who speak in binary, and we're sitting in between them. What have we got to do with them? I mean, why would someone working with genomics be sitting on a conference like this? Is it what? Do we do anything useful with regards to you guys? How do we help you, the binary speakers?
So I mean, I'm not a genomics person. Thiago is, but I'll tell you what I experienced from this thing. So the link I have with the healthcare guys is that it's like they collect data at multiple locations. That location is the key for me. So when I'm trying to put this information at different locations, now that's where I have to create insights from, because I was startled actually when I first saw this data 3 years ago, almost 3 years ago now. Every genomic sequencing data that comes out is at least 6 to 10 gigabytes. One sample gives you 6 to 10 gigabytes of data.
Environmental sample.
Yeah, and there's no way in the world a human can sit and go through that one data point even in a month's time. So you have to employ computers to derive insights from that. And there's so much information in that, and there's so much material in that, right? Every small characteristic of whatever you're observing is in a small part of that thing, and it is different for different species, different
DNA fragments.
DNA fragments, and different levels of species, basically, right? There are two elements there. First, for me, was overwhelming. First of all, there's so much data there, and I don't know how the bioinformaticians deal with that thing, but anyway, they deal with this, and they come up with very good insights. Now, that's one level of AI. Thiago will talk about it more on that thing, but then now you get certain level of information which is already features that are derived from this huge data set. Then we add the element of spatial locations for the data, right? Now I need to look at more features where I can look at correlations, spatial correlations and correlations to other variables at that location, and then you have time factor as well in that thing, right? So you have time, space, and in this case space is 3 dimensions as well, location and also depth in the water or height wherever you're looking at, right? All that information is so huge and the patterns are so diverse, there's no way a human can sit and look into that thing and make some insights or derive patterns from that thing. This is why AI is important, because we teach the AI on examples of what I'm looking for, and when you throw this whole bunch of data at it, it lets me see what those patterns are and where you're finding them in that entire stream of data, and based on that you can derive certain insight which is essential for a problem. For example, it can be a biodiversity estimate, or it can be an effect of different climate or land cover or land use patterns on that biodiversity. It can be an effect of fishing in this area on fish in that area, or if mangroves are affected because of climate change, what is happening to the fish population. These are all spatial insights we derive from the patterns we get from genomic data.
So we've had a journey together for nearly 4 years now, trying to figure out what we're trying to do. It's a very complex journey, and it starts by some sort of asking a question. And the questions that we're trying to ask here must have a purpose. So there's a lot of people developing a lot of software solutions here. Some of them are here to generate revenue. But what is the main purpose of everything we're doing now? Why are we spending so much money having to go and provide business cases to give us the biggest servers on the planet?
Why?
Yes, why? Generating so much heat and a lot of cost. There must be someone's paying our salaries for a reason. So what are we trying to do?
I like to say that I am a problem solver. Give me a problem, I will try to give you a solution. That's why I like to answer with that question. So there are multiple problems in the world today. I'll give you one example. By 2050, the literature and all the scientists in the world are saying that the antibiotics are not going to be available anymore, and millions of people will be dying because we don't have antibiotics to cure these people from infection. So talking about insights and trying to answer your question, we are trying to solve problems of the humanity today. From the data generated from the project we did on the ocean, and the current project doing on plants, and another one we are doing on soil. We are trying to gather as much as information from what is in the environment. I'm talking about virus, bacteria, fungus, and the other microorganisms over there. Understand the genomic properties of them, understand the weather conditions, understand the pH of the ocean, the pH of the soil, understand the humidity of the soil. Put all of this together, and the idea in the end of the day is solve a problem. Which problem? For example, one of them is the AMR, antimicrobial resistance that we have today. For the last two decades, I think, we don't have a new antibiotic in the market. Fortunately, AI today in 2018, one company developed one antibiotic using AI, but as you used to say, Dr. L, the responses for our problems are in the nature. The nature is our library of responses. So understanding it and solving this with that resources, I would say what we are trying to do.
So I think what you've touched a very good point about our relationship with the environment. We tend to forget that, nature is like IKEA. If it finds a good trick, it repeats it again and again and again. So there isn't much difference between the proteins we have as humans and the proteins you identify in the environment, whether it's in a bacteria, in a banana, in a plant. So there's a lot of similarities, because DNA is just a code for everyone here who does, the binary speakers. It's a 1 and a 0. We only have 4 numbers, so it's not a binary number to speak. Which brings us again to, why am I sitting with someone who's been working with satellites? Population genomics and a pathologist. It's like a joke, and 3 people work walking to a hub. So, and I'm sitting in your desk at the moment, where you were sitting 10 years ago.
Yeah.
So, how many things have we found so far?
Now, one of the first things, it's like I use the fish example because that's one of the most important things we did as a starting point. It was an amazing collaboration actually. It's like it was for the first time a full fish genome sequence was sequenced on board a ship, and on a moving ship in a way.
Yeah.
You know what that means basically. So I use this example of an aquarium. So let's say you have a huge aquarium, and there's certain fish there, certain all aquatic animals that are there in the aquarium. The way they explain genomics to me is the environmental DNA process, especially is if I have this huge aquarium with so many different aquatic animals in that thing. They told me, you know how important genomics are? You just go take a water sample from that aquarium, go to the lab, and we can clearly tell you exactly how many different animals or different fish or different aquatic animals there are and what type, right? And if we are lucky or if we are strong in AI, we can also tell the health of each of those, health status of each of those things, and potentially age of all of those things as well, right? That's so much information from a simple water sample. Now imagine scaling this up to the ocean or to the waters, getting these samples at different locations, right? And taking that entire thing and creating value out of that by just simply collecting water samples in different parts of the world. I don't know if I'm losing track, but I'm excited about this topic. That's why I can speak. I'm not a biologist. I'm a guy who builds satellites, and I got excited. Now there's so much more information you can add to come back to your point. You're collecting all this information. You're getting so much insights on the life that is there at different locations. Now satellites give me other information, right? The status of the water, the temperature of the water, salinity, right? Or other factors that are going to affect, anthropogenic factors that are affecting the water. For example, if you're living in Abu Dhabi, the population is growing, you're continuously feeding minerals through our waste into the Gulf. Now that is creating a lot of algae blooms. Algae bloom is other matter in the water that is damaging the fish populations. The mangroves are getting affected, which means that the breeding grounds for the fish are being affected. There are a lot more things that are happening. This is what we observe from some satellites and other data sources. And when we combine the information that we're going to get from samples, genomics, water samples and genomic samples, and the information that comes out of genomics, and you combine the information we are putting together from the satellite data, from other data sources and so on, we get this holistic picture of what is happening in nature. The same applies for biodiversity, for instance. Biodiversity loss is, you cannot really measure it. It's humongous. You cannot measure it. Doesn't mean that you should not be measuring it, but there's no proper way of measuring it because it's so much of value. You cannot put a dollar value to that thing, right? Natural capital is so huge. Now, I can observe from space what is happening in terms of land cover, land use, and all those factors. Now, if you go and continuously, repeatedly check what type of biomes are being protected, or how the biome is changing in a location, if you take this data, if you infer what is happening on the surface, and then relate both of them together, there are a lot of insights you can derive from that thing in terms of not only protection, also about understanding of the evolution of certain types of things as well, right? So, for example, honeybees got evolved so much that we are losing a lot of honeybees, but some of them got adapted as well. They got adapted because they were finding a right balance in the changing environment they could survive on, right? What is the changing? We are measuring the changing environment from the satellites. And the genomics and all this work is going to help understand the changing properties of the bees there in a way, If you relate that and figure out a solution, a practical solution of how to protect them, that's going to save the world. This is our mission.
So it's a pretty big statement. That we're going to save the world. Is the ambition. It's a pretty big ambition.
Yes.
And it takes steps. I think the first step is realization that you need different people from different experiences, from different walks of life, who see things differently, supported by the tools that are available all around us here. With a function and a purpose, apart from revenue generation, and having a question to ask and answers to fulfill, because the world is changing very quick, much quicker than we think. Which brings us to biodiversity conservation. It's a double-barreled world. Only biodiversity? Does that include anything apart from bio? Is there any other type of diversity that we need to conserve? I mean, is cryptocurrency diversity important for us? I don't know.
Yeah, I would say that the word itself is already explained. Bio in Latin meaning life as a complex thing. So we're talking about the land and also the ocean. Today there is a concept in biodiversity called bioeconomy. So we are talking about bioeconomy in the land and also blue economy that is refers to the ocean economy. The use of satellite plus genomic data brings together. I'll give you another example. Not mentioning the country, but in the carbon credit space, there is a lot of mislabeling or misconception on that. But the use of satellite images plus genomics, it's a powerful tool to do the monitoring, surveillance of a plot of land was put in a carbon credit market, and the genomics can be the tool to oscillate, I would say, the satellite image to say that plot of land that's worth X millions or billions of dollars has an increase or decrease of biodiversity because humans are interfering on that. The same concept applies to the ocean as well. We are developing over the last 2 or 3 years algorithms in AI and in genomics and bioinformatics to monitor and somehow certify, we are not there yet, but we will reach, the use of genomics to say the biodiversity of that area here, it's increasing or decreasing. That concept I'm referring to increase the amount of life in that plot of land. Why this is important, just to finish. In the carbon credit space, there is a concept of selling an area with eucalyptus. So I reforestate that area with eucalyptus. Eucalyptus will kidnap, sequestrate a lot of CO2, yes, but all the other vegetation, all the other animals will be dead because it's so specific, so much things to keep only the eucalyptus that you lose the other ones. So the concept of biodiversity today in that space brings together the idea of, okay, let's reforestate, let's preserve, but the whole ecosystem, not only one.
That's a very good point, actually. Yeah.
So based on the insights
the fact that we're working with non-human beings in Genomic and we're not working with humans, where do you think the space is going to move to, and what are other exciting projects do you think we're going to start in the next year or two between the two of us?
It's for me or for him?
No, both of us. No, it's like no. This combination is becoming very powerful. The biodiversity domain is huge. This example is so powerful. So there are examples. So when people were trying to reduce the carbon emissions, for instance, they were going and planting things, right? Mm-hmm. Without even thinking about. So you're increasing the forest cover definitely, but without ever thinking about how this is affecting the local biodiversity or bioregion. Eucalyptus is a very good example. When you're not planting the non-native plants, native plants, if that is non-native to you and if it is not significantly influencing the local biome, you're actually wasting it in a way, even if you're increasing number of trees, right? That's a very powerful example of how these two worlds should work together in a way. But there's a lot more things. It's like you have land, water, and atmosphere, right? So all three elements on land, you're checking what is happening on how the land surface processes are changing, what is happening with the land cover, how is the land used, what is happening as a response to the natural disasters, and then affecting and going and checking what is actually happening to the biome, from the microbes all the way to the life, to the plants and animals. Then on land, on water, in water basically, what is happening exactly, because land is at least localized. Water is now globally related basically. The water circulations happen globally, So many things are affected because of all the circulation patterns and exchange of material and all those things. Anthropogenic influence on water is super high these days because of our own ill doings basically, right, or whatever, Now that's where you need to really understand how this macro macro factors are affecting the micro factors. You need information from both sides. Then atmosphere. Now you don't know how much biomaterial is in the atmosphere, by the way. It's not just dust and oxygen and nitrogen. There's so much of biological material in the oxygen, and you really need to understand how this is done, because this entire thing is transported thousands of kilometers. The dust from Sahara reaches here, dust from Sahara reaches Europe, dust from Levant region is coming as storms into this region, right? Through this dust, there's a lot of biological material that is getting transported as well, right? You need to be able to understand what is happening in that entire transportation mechanism, what is changing, what type of new microbes are introduced into these ecosystems and so on, right? It's an exchange, it's a continuous process. So in all three domains, land, water and atmosphere, we need to work together and build a new narrative of what is happening in nature.
So for closures, Tiago, we've had this conversation many times with regards to what we call the currency of measure. Is there any currency to say that the UAE is more diverse than Amazonia or less diverse? What is it based on? Why do we say that Amazon is? What's the currency that we measure at the moment?
Tough question. I would say the currency is biodiversity itself, but in words terms, if you take a micro-environment, maybe this place here has a higher biodiversity than this other place here. What's the currency? Biodiversity is the currency, Dr. El. And I would say we are not there yet, but we will reach very quickly.
I'll correct that, biodiversity is the label of that currency. But there's no quantification done yet, and that quantification has to happen. And that is when you actually create a value to that label of currency in a way.
Yeah, and going further down that, what we call these sites in no human genomics is not only we are talking about health, there is the unhealth space as well. We have a laboratory in UAE called RACID that also monitors the health of the environment, outbreaks of bacteria and pathogens that are out of there. Just to go back to you, genomics, satellite, image satellites, and AI are the enabling, in my opinion, to allow us to quantify biodiversity, and I think this will, in a cascade word, enable us to have a proper answer in maybe 6 months, 1 year.
More, probably.
I'm an optimist.
6 months. So with that, I think we probably close this session. And thank you very much for your attention, and we hope you found it interesting. Thank you very much, everyone.
Thank you. Any questions?
Yes.
Yeah.
Yeah, go ahead. Sorry.
Get the microphone.
Use this. Use this.
Use this.
Thank you. Amazing session, yeah?
Thank you.
Loved it. I'm no scientist, not even close. I may have a couple of questions. Stupid only, don't call it out. The first one is, you said sample collection of genome sample, or rather, sample water, and then you do all your analysis. So, how does one validate that authenticity of what you come out of? Now, what do you bring out? This is one. First question. Second question, what was it? I forgot. I tried to remember, but you can answer the question.
So you can answer that, but I'll tell my answer, then you can validate me. So you validate all this works in controlled situations, basically. In a lab, in a controlled environment, you validate both the results first, in a way. No? That's how you do it on multiple samples. Both. So this is a sampling 101, basically, statistical sampling 101, where you have to collect a number of samples, validate it in different environments as diverse as possible. Once that validation happens, then you have a proof that this is going to work. Once you are done in the lab environment, then you go into the field environment, you revalidate based on traditional approaches, like the trawlers and all those things. That's why when they did the first time, that was a huge task, because we were following the local government fisheries survey, and exactly where they put the trawlers in and collected fish, we also collected water samples. And they went through that's why sequencing the genome, processing that entire sample for environmental DNA extraction and sequencing that on board the ship is very important, because I cannot take everything to the lab and compare that, right? So this was the first time all over the world that this happened on the moving ship, and that is a way to validate them together at the same location at the same time, so that we enforce that this is suitable for future surveys. We're not there, but once we establish this thing, in the future they don't have to go through these huge trawlers, catch thousands of fish, break them, make measurements and all those things. Instead of doing that, you can go in the same ship in a smaller one, collect water samples using those Niskin bottles, and then put it on the lab which is in the ship, take a sequence, send the sequence to the cloud, then you get all your answers. Am I right?
Yeah, absolutely. From the same place, we collect the fish, we collect the water. We sequence the fish, and in the water data, we look to the DNA of the fish. And we are able to find it there. Of course, took a lot of time and resources to build the pipelines. The bioinformaticians behind of us who did the work, they had a lot of headaches, I would say, to find out the right algorithm to do it. But we managed to do it.
Thank you. One last question. So how do you handle bias when you're writing your algorithms? So human bias is always a problem, right?
Yeah,
So how do you mitigate that?
especially with the genomics data. If you have a 6 to 10 gigabytes data stream of one sample, then definitely will be biased. Because that's where the reference. Sorry, it's breaking up. I'm getting excited. Anyway, sorry. That's where sorry. Can you hear me? Okay. No. Let's break this. It's okay.
I'll take part of the question until he gets mic'd up. So when you talk about bias, there's particular statistical methods that you use to reduce the probability of bias from the experimental design onwards. To make it simple, you start with the simplest experimental design that can confirm what we're testing is what we're testing, and that starts by working with a smaller sample in a controlled environment. There's a lot of areas of bias and failure that you can have even before you generate the data. Yeah? So even before generation of the data, there's going to be a lot of areas of bias. We performed a lot of pre-experimental design in controlled environments. One fish in a goldfish in a goldfish tank first, basically. We see how much environmental DNA we can get, what she sheds, because she also sheds not only her DNA, but a lot of her gut microbiome DNA, and we have to go and put that in pipelines. First of all, have we been able to identify segments of the DNA that are particular, and then remember that these are only small segments and fragments. So we start working all the way from kingdom. Is it a fish? Yeah. Then we go down to genus. Is it from that genus? Yeah. And to go to species is very difficult. We're not there yet, and we cannot assume. But we have identified. We've reached to fathom for data at the moment. I mean, one of the biggest things that we have to clean for is human DNA in the ocean. Remember when we were trying to figure out what the hell is this? The biggest chunk of DNA we have to clean is actually human. Yeah, not fish. When we're subtracting the DNA to try and figure out what we're getting from DNA, believe it or not, there's much more human DNA in the oceans at the moment from environmental DNA sample in the most isolated locations that you can imagine. And we found another experiment that we did was really, you can probably find it on the internet, the microplastics one. You know, that was we did that. Is that published on the website?
Yeah, I mean you can see, it's on the website. Yeah.
So we were collecting just 1 milliliter
10
water samples.
10 ml of
Yeah,
water.
it started with 10, but then it came down because. But you can tell the stories and go back to the bias.
Just to give you the data scientist answer for the bias. There's always a reference library, and we continuously improve the reference library. So every single time you do something, you go back, check with the reference library for specific features that we extract. You don't have to check for the entire 60 gigabytes of data. We need to compare the fragments, the DNA fragments that we extract for that thing with the reference library, and then we repeat that with everything. So that will answer your data analysis part of the bias in a way. I was talking about the other biases, but anyway, on the microplastic side, we had a very interesting story. We were sponsoring a sailor in a race called Golden Globe Race, and Abhilash was here yesterday. Maybe he was around here anyway, and he was going around the world in a sailboat, no power. Yeah, and what we did was we took it as an opportunity. The opportunists, the scientists between us, we gave him some empty 10 ml water bottles to collect water wherever he goes. Right?
Yes.
We got back the water after 10 months at sea. Basically, he was alone in a boat all over, in a sailboat powered by wind, by the way. There's no power, and he has to take all the food with him for 10 months. And survive on that and go around the world. So now he came back with that data of 10 months with water samples collected over 100 locations, and his boat flipped a couple of times in between in the sea. The samples were recovered. Then we got back, he got back, gave us the samples. I gave it to Dr. Weil, and he was supposed to tell me what is in that water samples, right? Imagine that 10 ml bottle is so small. Sometimes in this entire thing even was dropped and there was 5 ml or less and so on, right? To infer, and then the biological analysis could not be done because it's 10 months in that bottle, right? You could do some other things like microplastics was a very good example, right? So we use that thing, new methodology by the way. I was surprised that they came up with that methodology to infer microplastics in that water samples. Microplastics are so small, they'll still be present. The insights were so surprising, and I would not put a sad tone there for now, but it was very sad when I saw the results. In parts where you are not expecting plastic, there was huge amounts of microplastics. And what these things do? They enter the fish, and they enter our food cycle. I don't know. There's more to that. Now that type of analytics could be done with these samples, right? And you can build on the top of that, infer some more features based on the genome and on the changes in the genomic patterns of those fish in different species, based on what is available in that location, right? These plastics are not just staying there. If the fish eats it, at some stage, 3 or 4 generations later, some things get mutated or some things get the DNA changes.
There is work showing that the microplastic can influence the gene expression, altering the pattern of expression that could cause disease.
Exactly.
Generations after.
Yeah, so these type of studies have to be done as well, right, along with this. That's where it's not only satellite data and genomics, but also all other data that could be collected to infer what is happening with nature.
Make use of a lot of the NVIDIA processors that are being produced at the moment. So a lot of what we do uses a lot of data. So we have a lot of very smart scientists. We develop methods in again, utilizing much of the computational prowess that is provided by computers' abilities. And the method that we use, for example, we were using self-sorting technology that's usually used for other purposes, but at a higher level with image recognition systems. And we had a lot of our AI engineers looking at the background to try and give us the right images for collecting. So I think, to cut a long story short, there's a lot of data all around us. Data is not just limited to the gaming or to the products or to the apps or to the folders. Or there's a lot of data in this room. There's not only facial recognition systems, not only the air we breathe, what we excrete, what we eat, what's the product of that. And this all is mathematical in one form or other. And hence, we're trying to combine the biology that we're working with the engineers from physics, with the molecular biologists here, to try and make this more a hospitable place for us, our children, and those to come.
We are good.